Good day, everyone. My name is Tshepo Nkuna, and I'm a researcher here at the CSIR's Chemicals Cluster. I will be talking about nanomedicine in the context of COVID-19. Although we do have data and everything, unfortunately, because of IP issues, I will be using a more similar model or a more similar disease that we've worked on. And basically what we're trying to show is that this is what we can do as well for the current treatment drugs or the drugs that are being investigated for treating COVID-19. I'd like to start by firstly acknowledging 360 Biopharmaceuticals, Totoma, and CSIR. This project was initiated in March, whereby all the stakeholders and our CEO with ESCO joined me in a presentation whereby I presented our concept idea on how we can tackle COVID-19. Secondly, I will speak briefly on how COVID has affected the world, and I think everyone should know by now that over 1 million people have died. On the 10th of October this year, over 8,000 people died. And if you look at the trend as of April, the highest has been 12,000 a day. Now, this virus that's causing COVID-19 disease, it's said to be zoonotic. Virologists say that probably came from bats. It has about 96% similarities, the genome, and then it spread through respiratory droplets through coughing, sneezing, or touching bodily fluids of infected people. And the symptoms, anything from 1 to 14 days, and the average symptoms onset about 5 days. I'm not going to talk much about the virus itself because I'm not a virologist. So infection is said to, the target cells for infection by the COVID virus, which is SARS-CoV-2, is the AT2 cells. They're alveoli type 2 cells, and there are two types of cells that make up the alveoli, which is AT1 and AT2. Now, the AT2 cells, they are crucial in maintaining homeostasis of the alveoli. And the virus attaches through the ACE2 receptors and gets internalized through endocytosis, and the genomic material gets released, gets transcribed, and then more viruses are made, and then they're released into the circulatory system whereby they affect other tissues and organs. Now, this is just to show you an overview of the tissue that express AT2 cells. So from your lungs, it can go to your small intestines, your brain, your heart, your eyes, almost everywhere. So yeah, it is a huge problem. Now, the symptoms, normal common symptoms that everyone or most people experience is the fever, the dry coughing, and fatigue. There are some uncommon ones, but the severe ones, you will have your high fever, coughing up blood, kidney failures. So basically, in general, it can also lead to death. So we all know that. Currently around the world, there are a few drugs that are being tested against COVID-19. Not a few, a lot. And there are those that are front runners, like remdesivir, which was developed by Gilead, initially for Ebola. Somehow it failed, and now they're trying it for COVID. It is an adenosine triphosphate analog, meaning that it is a more of a replica, but not that similar to the adenosine itself. So what it does is that if the virus gets into the cell and releases its genome into the cytoplasm, now coronavirus, its genome, it's a positive sense RNA. It's positive sense RNA genome, meaning that it does not require to go inside the nucleus for it to be transcribed. It can do that within the cytosol. So now what happens is that remdesivir goes, after it gets absorbed, it has to go under phosphorylation. So there's a cleavage that occurs and it gets phosphorylated so that it becomes an active metabolite, an active analog. So when the genome of the virus gets transcribed or gets replicated, if remdesivir gets to bind or if gets to be included in the replication process, it causes an early termination sequence of, so more nucleosides cannot be added. And there's also chloroquine and hydroxychloroquine, but these ones, they don't have a direct mechanism of action, so it's an indirect. So what happens is that chloroquine and hydroxychloroquine are highly absorbed. And when they get absorbed, they get entrapped into these small microorganelles, we call them endosomes, just small compartments within the cell. What happens is that when that occurs, anything that goes into the cell through or via endocytosis, now it forms a phagosome. Now that phagosome gets attached by a smaller organelle called, it's containing lysosomes. So the fusion of that forms a phagolysosome. Now the phagolysosome, its mechanism of action is that it decreases the pH. By decreasing the pH, now everything gets degraded and then gets excreted when the phagosome and the lysosome fuse, they form a phagolysosome, whereby the pH inside goes down and everything gets degraded. But now, if chloroquine gets entrapped in a phagosome and the lysosomes come to form a phagolysosome, it alkalizes the environment. So it stops the process of acidifying the environment, and that's how it works for malaria. Unfortunately, there's a few resistance, but now there are still investigation of the exact mechanism of how chloroquine works. Dexamethasone, lopinavir, ritonavir, they are antiviral drugs, and viruses have the same replication mechanism. They insert their genome, and then the genome gets replicated to form more virions, which then gets released to infect other cells. So antivirals, by incorporating them in the chain that's being synthesized, or the genome synthesis, it breaks down the replication process. Now I'm going to focus a little bit on remdesivir, and that's a typical structure of what how remdesivir would look. And as I mentioned, it's an analog of adenosine, which was developed for Ebola. So just to stress this, it's an FDA emergency use. Currently, the recommended dose, it's about 1.1 gram divided into 10 days. The first dose or the first day is 200 milligrams, and the subsequent days, which is 100 milligrams. Now, this is given via IV infusion. And IV infusion, not just a needle and injection. So what they do is that they have manufactured a lyophilized version of it, meaning that they formulated it so that it kind of solubilized in water. I think they're using 0.9% saline. Now, the treatment duration for one dose, it's about 2 hours, 30 minutes to 2 hours, anything from 30 minutes to 2 hours. Because of how they do it is that they mix the lyophilized powder with the saline, I think about 100 milliliters or 250 milliliters. Now, it's a drip. They inject it, and then throughout the 2 hours, you just continuously receiving the drug. Now, the problem with that is that it is water insoluble, meaning that it is oily-like. And as we know that our body is like, 90% made out of water. And then now the problem with highly insoluble drugs is that as soon as they are taken up into the body, they get excreted immediately or get degraded by the liver. There's a whole lot of processes that occur within the body that make sure that oily things are removed or stored or just moved out of the way. Now, back when they were testing remdesivir for Ebola, they used primates, I think monkeys or baboons, and the half-life was about 40 minutes, meaning that if you take it now, half of the dose will be lost within an hour. The biggest issue I think that we as South Africans, if we were to import remdesivir to start using it, is that it is very expensive. They are saying about $520 per dose. Now, this is where nanomedicine can play a very crucial part. More than half of the drugs are current treatment therapies for almost all of the diseases. Is that the issues is that you have to get a repeated dose, mainly for the fact that the chemical nature of compounds or drugs that are used to treat, they are more hydrophobic, meaning that they are more oily. And like I said, the body doesn't like oily things. It takes out oily things. So I've listed a few of drug limitations currently in market. Your solubility, which is the biggest issue, toxicity, low efficacy, efflux pumps, and your hepatic metabolism. Now, this is where nanomedicine comes in. Now, these limitations can be mitigated by employing or using nanomedicine. Now, nanomedicine is a branch of nanotechnology. As the name states, is nano meaning numeric scale, centimeter, millimeter, micro, then nano. So basically we are working at that level. There are various applications that nanomedicine can be applied to. And then in our case, what we do is that we encapsulate drugs and use conjugation, meaning that we develop these small capsules, spherical. I'll show you a picture now, whereby we literally incorporate the drug inside. Now, the surface of these particles, they are hydrophilic, meaning that they like water. So if you put it in the body, the size plus the hydrophilicity of it makes it easier for the body to absorb. And there are different pathways that nanoparticles can be absorbed. And we've got your lipid-based polymer, your drug conjugates. Now, your drug conjugates, it's where you take a highly hydrophilic molecule, meaning a polymer that likes water, and then you covalently add, so you chemically link it with the drug. So you would need your drug to have a functional group like your OH, your amine, and so on. The materials that we use, they are biodegradable and biocompatible, meaning that when you take them into your body has the ability to break them into smaller monomers, and then it can use them for other things or just excrete it easily. So there won't be an accumulation of material in your body. We can design systems that release drug at a prolonged duration, and it also minimizes toxicity, your adverse side effects and all that. Now, a picture on your left, on my right, is a typical example of how the distribution of nanomaterials will be distributed throughout your body. And the benefits, as I mentioned, is that it will increase, by increasing the absorption of drug, you're also increasing the availability of the drug or the bioavailability of the drug. You can also bypass the first pass metabolism, meaning that when your drug gets absorbed into the small intestines via the hepatic portal vein into your liver, you can protect your drug from being degraded by the liver processes, the cytochrome P450 enzymes, and reduce the dose and the frequency as well. So the graph, what you see there is that the zigzags, the up and down, the wave on the graph is that it will represent how an individual will take medication on a daily basis. When you take on day 1, the graph goes up, but reaches its peak, and then it goes down. So tomorrow you have to take it again, over and over. But now with our system, you take it once, and throughout, you receive enough dosage above the MIC, meaning the minimum inhibitory concentration. And I will just show you now. Like I said initially, that I won't be talking about exactly what we're doing on corona. I will talk on a drug that more or less works similarly, but not that much, but it's a close enough of an example. The difference between a virus and every other pathogen is that viruses release their genome or the genetic material into the cell to make more copies. But other pathogens, what they do is that they duplicate. So they don't need the machinery. They don't need the machinery of the cell to make more copies. They just make copies. They just divide and make more copies and infect other tissues. The difference here with SARS and TB, their infection, the site of infection is the same, the alveoli, but there are different types of cells. SARS infects the AT2 cells, and then the tuberculosis infects the macrophages. But SARS also can infect the macrophages. Now, I will be talking on rifampicin. Rifampicin is more or less similar, or the dosage, the solubility, and the half-lives and everything. So the picture that I was telling you about, how after we encapsulate them, they will typically look like that picture. So rifampicin is also highly hydrophobic. It is highly bound by proteins as soon as it gets into the circulatory system. And also the half-life is decreased when co-administered with isoniazid because of they interact in acidic conditions. And there's a few other issues. Now we have encapsulated rifampicin, and then we've infected animals with TB, and then we treated them with the conventional treatment versus our treatment. And in the lab, before we went to animals, we did a release of the drug within these capsules over time in different physiological media, meaning that we will do it in your simulated intestinal fluid, your simulated gastric fluid, and your PBS. So basically, it kind of gives us an idea of where and how and for how long the drug will be released throughout. And this graph shows that we managed to release drug for over 20 days. And unfortunately, I can't give more information, but it's looking good. It looked good because of then we went in animals. And then in animals, this is what happened. So your blue, the first graph, your blue with triangular will be your conventional therapy for TB, meaning that will be your day 1. You take drug, it goes up, then it goes down. Now the light blue line is the MIC line, meaning that you always have to be above that line so that you get treated. So that is the minimum amount of the drug that you need to be effective against the pathogen. Now the other two lines, now that's a single dose of our encapsulated rifampicin. So as you can see, even inside an animal, we managed to sustain the dose above the MIC for over 100 hours. So that was good enough for us to go for efficacy studies now, meaning that we go into animals that are infected with the actual tuberculosis bacteria. Now, this is what happened. The distribution of the particles, they were everywhere. Brain, heart, kidney, liver, lungs, spleen, plasma. So The 3 bars represent 3 formulations, and yeah, IP issues. Now we went on to do efficacy studies in diseased animals, and we compared the conventional versus our drug delivery system with rifampicin. Now, the conventional therapy, we kept it as is, once daily, but for our delivery system, we only treated once. We gave 1 dose. Now, what you see there is the results of a single dose of encapsulated rifampicin versus a daily dose administration of the conventional therapy. Much as the current one performed better, ours also showed promising results. And with the current investigations that we're doing now and a few other ones, we have shown, or we have managed to reproduce these results. And lastly, this will be your lung pathology of the diseased animals. So now the small bubbles that you see, those will be your granulomas, your granulomas whereby the TB resides within your lungs. Now, if you look at the timeline of the rifampicin gave daily, the current therapy, versus ours, there's more of a similarity, meaning that there is efficacy. So our system works, and over the years we have perfected it, and now we have not started, but we have already started working on some of the drugs that have shown to work or be efficacious against COVID, and we've got some exciting results, and you'll hear about them in a few coming weeks, months. Thank you, and now we're open for questions.